The overall goal of the following experiment is to monitor endoplasmic reticulum calcium dysregulation over time using a secreted reporter protein. This is achieved by viral vector-mediated transgenesis of the target tissue, specifically liver in this example, with the Gaussia Luciferase SERCaMP reporter. Next, biological fluid, such as blood, is collected in order to measure the amount of SERCaMP released from the tissue. Then, the luciferase substrate is added to the plasma samples, and luminescence is measured to determine ER calcium depletion. The results show ER calcium dysregulation as indicated by increased levels of plasma SERCaMP. ER homeostasis can be monitored over extended durations by repeated sampling of small volumes of biological fluids.
The main advantage of this technique over existing methods, like low-affinity calcium dyes or genetically encoded calcium indicators, is that ER calcium homeostasis can be monitored in vivo over time via a minimally invasive procedure.
There are two therapeutic avenues for this technique. The first is to screen for drugs that can maintain or restore ER calcium homeostasis to normal physiological state. The second approach is to modify therapeutic protein to become therapeutic SERCaMPs where their secretion occurs in response to ER calcium depletion.
Though this method can provide insight into ER calcium dysregulation in the liver, it can also be applied to other organ systems such as brain, heart, muscle, or pancreas.